Hello, I'm MELİSA KALKAN.
I'm TÜZÜN.
We are 11th grade students at Diyarbakır OKYANUS High School from Turkey. As you know, it has become more important to use biodegradable films instead of plastic packaging and films, which cause environmental and health problems. We aim to produce a natural and antimicrobial biofilm on our project by producing biodegradable films, Quince extract. We aim to extract the shelf life of the foods and offer an alternative to use of plastic, which is a major environmental problem today.
Quince is a sticky and tasty substance, so we aim to make a biofilm with natural and antibacterial properties of quinces. In our biofilm, we've used quinces, which are vast and unusable. In previous studies, it has been stated that quince is a source of phenolic acid, antioxidant, and flavonoids. Based on these features, we wanted to utilize this waste in our project.
In our study, we first extracted quince extract in the laboratory and used a biofilm production stage.
Before making our biofilm, we performed antibacterial analysis with the extract we obtained from quinces. In order to determine its antimicrobial effects, we applied the agar-well diffusion method with nutrient-agar medium. After 48 hours of incubation at 37 centigrade degrees, we measured the zone diameter around the well. As you can see in the table, these results show us that the quinces extract has a high antimicrobial activity.
We started to make our biofilm after antimicrobial studies. So how did we do that? For biofilm production, we mixed agar, gelatin, distilled water, and glycerin in a magnetic stir at certain temperatures, as you can see in the figure. Then we divided the prepared film solution into two separate beakers and added quince extract at different concentrations respectively, and mixed for another 5 minutes. We poured the film solution we prepared in thin layers into petri dishes, previously stripped with UV light, and left to dry in the film hood under room conditions.
As a result of our experiments, we obtained a high quality of biofilm from quinces. We conducted trials on cheddar cheese samples in order to determine the food preservative properties of our biofilm without any additives. We formed 3 groups of cheddar sample to be used in study as uncoated, 100% quinces extract, 15% quinces extract. We kept the samples at 4 centigrade degrees for 15 days and performed both visual microbiological analysis every 3 days. According to data in our visual analysis, the uncoated cheddar cheese sample started to dry on the 3rd day, started to deteriorate on the 6th day. The best results was obtained from the film 100% quinces extract. This shows that oxygen barrier formed by the film is effective and has an edible film feature.
In our antimicrobial analysis, we first took samples from the control and experimental groups on the 5th, 10th, and 15th days, and we performed a pre-enrichment phase in potent water for the growth of microorganisms. Then we prepared a deletion series from these samples. By taking 100 microliter from each sample, we applied spread plate method to microbiological medium taking nutrient agar in a sterile environment for bacteria count. We kept petri dishes in a 37 centigrade degrees oven for 48 hours, and at the end of the period, we counted the bacterial colonies that live in the medium and evaluated their density. As you can see in the table, when the colony count results of the film-coated cheddar cheese samples with the extract prepared are compared with each other, the best result was obtained in a film-coated 100% quince extract film.
To sum up, when we evaluate the results of our study, we can say that our film coating containing quince extract can be easily used in the food industry by using some in progress. With edible films to be produced, it will be possible for the both industry and the food sector to develop in this area. It will be an industrial benefit for the future studies to include scoops to reduce production costs. In addition, we think that our project will contribute to health packaging of food in a healthy way and deliver the consumers within the scope of COVID-19, which the whole world is struggling with. We think that our project will contribute to healthy packaging of food and reach the consumers safely. Thanks for listening.
Haydi ettim.